Hello, my name is Luke Harris, and this is my video submission for the bonus points for Professor Shahid Mukhtar's Genetics BY210. In this video, I will go over the steps of DNA replication, DNA or RNA transcription, and translation. Basically, all the steps that will get you from a DNA strand to a polypeptide that will make a protein. So let's begin. The first process I will describe to you is DNA replication. DNA replication involves the duplication of DNA. In eukaryotes, there is multiple origins of replication. In prokaryotes, there's only one because it has circular DNA, and prokaryotes has linear. You have a primer, which is a signal for DNA to start replication. Helicase is the enzyme that separates each strand, origin of replication. Each of these strands either one of these strands can be a template. RNA primase adds RNA primers to each strand, and DNA polymerase III adds new nucleotides and creates new phosphodiester bonds. And as these strands go, this is the leading strand, which is continuous, and this is the lagging strand, which is discontinuous because of the direction of replication. These fragments are called Ozaki fragments. When these fragments need to be joined up and the RNA primer is removed, this is the job of DNA polymerase I. It removes the RNA primers, and then DNA ligase combines them together and makes a brand new DNA. The second process I will describe is transcription. You have an RNA-like strand, the 5' and 3', and you have a template strand, 3' to 5'. RNA-like strand will be exactly like your RNA, and template strand will be anti-parallel and complementary. Either strand can be the template. In prokaryotes, you have a promoter sequence of -35 and -10, which is essential for transcription to start, and you have a terminator sequence. You have RNA polymerase is a core enzyme. It cannot start until the sigma factor binds to it and makes a hollow enzyme. Once that starts, transcription can continue from 5' to 3'. Then you have a termination signal of a hairpin, which signals this core enzyme to stop transcription. And then you have your mRNA. In eukaryotes, it's a little bit different. You have an enhancer sequence, which enhances the speed and supports the transcription. It enhances the transcription, basically. You have a promoter sequence right here, which has a title box, which is a general codon, or not codon, but a general sequence that initiates the start of transcription in eukaryotes. And RNA polymerase II makes the pre-mRNA. It is the enzyme that actually makes the pre-mRNA, because eukaryotes has to be edited before it is a full mRNA. You have DNA RNA polymerase, which pulls this strand off. It is capped with a backwards G on the 5' end, and then on the end, after it is cleaved by ribonuclease, is cleaved and a poly A tail is added. So you have a 5' G cap and a poly A tail. Then you have introns and extrons. Introns and exons. Introns are not needed in mRNA, and exons are. Spliceosome removes all these introns, and then you have a mature mRNA with exons, 5' cap, and poly A tail. The process I'll describe is translation. It has an initiation phase where the small ribosome subunit binds to a 5' methylated RNA cap, and it scans down to find an initiator program, AUG, an initiator codon, AUG. A tRNA binds to this initiator codon, and the small subunit scans and sees that, and it has F-met or met, depending on if it's prokaryote or eukaryote. You go down, the large subunit binds on top of that, and it sandwiches this tRNA inside it, with a P site and A site and a E site. The elongation phase is where this ribosome structure moves down the mRNA, and more tRNA join these anticodons and codons. They join, and then these amino acids link to form polypeptides as it moves down, and they move through these chambers. Then you have your termination phase. The termination phase is where, as these amino acids are linked together to form a polypeptide, a release factor is joined. This release factor signals the stop of translation, and it binds to UAG, a stop codon, a termination codon. So in that place, the large subunit separates, that separates, the small subunit, the release factor separates, mRNA separates, tRNA separates, and then you're left with this polypeptide chain. It will go on to form proteins and perform essential functions within the cell. And that is it.